Welcome guys to BHCS 2006, the Immunology Practical, and this will be the Enzyme linked Immunosorbant assay. Before we even start, I've got to give you some housekeeping, and it involves safety. Please make sure you're wearing the appropriate safety gear, the lab coat, the gloves, because we are going to be using both caustic substances such as sulfuric acid, and we are going to be using cancer-inducing substances such as TMB, the colour reagent, later. So please make sure you're protected against these reagents. Now, supplied with this practical is a protocol, and we're going to work through the protocol that has been provided for you. You are going to refer to page 3, step 1, and you are going to work through it systematically. What we need to introduce you to first is that you are all going to do this individually, and you are provided with your own individual work tray. On that work tray, you have a pre-coated ELISA plate, you have pipettes, you have pipette tips, you have diluent, and you have the reagents for developing colour development to measure enzyme activity. So all the reagents are provided with you, and the step that we are going to start off with immediately is preparation of the standards and samples to put on the ELISA plate. And the standard will start by going to the tray, and you will be using this beaker. Okay, this contains 1 microgram per mil of HRP. Now this HRP stands for horseradish peroxidase, but the way that we've developed this ELISA, this is the antigen that is going to be captured on the ELISA plate. Okay guys, so what we're going to do next is create the series of standards and the samples A to D ready to load onto the ELISA plate. So we start off, we've indicated that this is the antigen at 1 microgram per mil. The top standard for the standard range is going to be 100 nanograms per mil. That effectively is a 1 in 10 dilution that you're going to set up. Bearing in mind the volume that can be taken in an Eppendorf tube, I would suggest that you use a total volume of 1400 microliters total in the Eppendorf. So 1 in 10 to a total of 1400 is 140 microliters of stock that you've brought from the antigen beaker, and you're going to dilute it 1 in 10 using the diluent, which is the PBS. So I've created that already, and you're going to create a serial dilution for the rest of the standards. And effectively, you add the appropriate volume, and you have to do these calculations. You add the appropriate volume of diluent into each of your tube, and then take from a previous tube and add into the second tube afterwards, and so on and so on, to create the standard curve range of 100, 75, 50, 25 and 5 nanograms per mil. So I've created 175. What I'm going to do is take out of the 100 and create the 50. So that's a 1 in 2 dilution. So nicely mixed. I'm taking out a certain volume and I'm adding it to an equal volume of the diluent, and I'm mixing in the tube. So there's 1 in 2 that creates 50. My next dilution is 25. I take out the same volume and add it to an equal volume in the new tube of diluent. Okay, now that is my diluent for 25 nanograms per mil done. What we need to do is finally do a 1 in 5 dilution to create the last standard, which is 5 nanograms per mil. So I'm going to take out 1 volume from here, and I'm going to add it to 4 volumes of diluent, your PBS, in your final tube to create that 5 nanograms per mil. And that is the dilution range created, ready to put on the ELISA plate, and you're going to put on these standard dilutions along with appropriate 100 microliters per well of your A, B, C and D, which are your test solutions. The next step, guys, is to add these standards and samples onto the ELISA plate. So we're going to add 100 microliters of each of the standards and each of the samples to an appropriate place on the ELISA plate that has been marked out in your protocol. Note, I haven't marked A, B, C and D. I've only marked C and D, A and B. So what you have to do is put C and D in the appropriate places. Note, the ELISA plate has only been coated from columns 1 to 6. Do not put any samples from 7 to 12. So I'm going to start off by adding the diluent. Now the diluent is effectively the 0 in the standard curve. So I'm going to add 100 microliters per well into F1 to 3. And that is the 0 for the standard curve. Now, because I'm starting from the weakest and going up to the most concentrated, I can use the same tip to add all the standards if I go from weak to strong. So the next one, without a tube change, is the 5 nanograms per mil, 100 microliters into each well of the wells above in E1 to 3. Next, I move on to 25, and that goes in D. Next is 50, which goes into C1 to 3. Then the next standard is 50 nanograms per mil, and that goes into B1 to 3. And then finally, for the standard curve, 100 nanograms per mil, 100 microliters per well, put into A1 to 3. That is the standard curve added to the plate. Now all I need to do is add the appropriate test samples A to D that you want to measure. So I'm going to add 100 microliters of each of A to D. Note, you do need a fresh tip between each sample, but I'm going to add 100 microliters per well in designated places on the protocol, or you can decide where to put them, so long as you put them anywhere in columns 1 to 6 on the plate that hasn't already had the standard in. So I'm going to put A, 100 microliters per well, in row A, 4, 5 and 6. For B, I'm going to use 4, 5 and 6, but I'm going to put B into row C. Then we come to adding sample C, and I'm going to put that into E, 4, 5 and 6. And then finally, I'm going to put sample D onto the plate at 100 microliters a well again, and I'm going to put this into G, 4, 5 and 6. Now that's everything made up. All the samples, all the standards, and they've been added to an appropriate designated place on the ELISA plate. Now the next part of the process is relax, chill out, we're going to incubate this at 37 degrees C on the hot plates that are on the other side of your bench. Okay guys, welcome back. We're at the end of the incubation period of this ELISA protocol. So if you refer to your protocol, we are now at the end of the incubation going to step 4, which is washing of the plate. So we take the ELISA plate and it's got your sample and your standards on. Now, a leap of faith here that everything is remaining bound to the plate. So what we do is we're going to flick the wrist and throw off the liquid. We're going to throw it into this bin, but we've lined it with paper to stop it splashing back on you. So be very careful, we don't want you to get covered in contents from the ELISA plate. So flick your wrist. There you go, that's the liquid removed. Now what we're going to do is wash the plate. It says on the protocol wash 6 times. You can actually wash 4 times and it'll be fine. This is where we're going to use PBS Tween in the wash bottle. The Tween is a detergent. It will wash off any unbound substrate that's left on the ELISA plate, leaving only the complex that we want to measure. So we go along. We're only going to wash half the plate because, as I said, only half the plate was originally coated. That was wells 1 to 6. So there's one soaking. I'm going to throw off again. That is the first wash, and I'm going to dab it to remove any excess liquid. Okay, that's the end of the first wash. I'm going to repeat that another 3 times. Two. 3. 4. So I'm going to leave the plate inverted there so that any excess liquid drains off, and in the meantime I'm going to prepare the colour reagent for the next stage of the ELISA. Okay guys, so you've washed the plate, it's draining. Now what we're going to do is make up the colour reagent, which is made in this citrate buffer. What we're going to do is we're going to add 100 microliters of TMB, tetramethylbenzidine. Please be careful with this, it potentially is carcinogenic. So make sure that you've got your gloves on and you're well protected. But we're going to add 100 microliters from the TMB in the Eppendorf, and we're going to add it into the citrate buffer that's provided for you. Now for this we've got to activate it. The enzyme that we're using is horseradish peroxidase, so to be able to initiate the reaction, we're going to add hydrogen peroxide for the peroxidase to kick in. So we're going to add 10 microliters to activate this substrate solution. We put the 10 microliters into the citrate buffer containing the TMB, make sure it goes into the liquid, and we're going to give it a bit of a swirl, gentle swirl. Don't shake it violently because we don't want you coating yourselves in it. Now from here we just tip the citrate into one of these ELISA troughs. Okay, that's good. Now this is where we now add the colour reagent to our ELISA plate in, if you remember, columns 1 to 6. So we're going to add 100 microliters using the microtiter plate here and add this onto the ELISA plate. 1, 2, 3, 4, 5 and 6. Now, if you actually have a good look at the ELISA plate, you can actually see a blue colour already starting to develop. That is the development of the standards. They'll create a standard curve to be able to quantify test samples A, B, C and D. So in standard operating procedures that hospital laboratories normally provide, they always define set times to be able to develop this. Forget all that. We are going to, as scientists, look to see how it develops in real time, and what we want is a discrimination in the quantity and intensity of colour between the top concentration standard and going down lower and lower to the lowest concentration standard. So that discrimination sets up a standard curve that we can then quantify our test samples A to D. So we're developing this until such time as we think is a reasonable time to stop its development. Now, what I can see already is that unfortunately there's not massive amount of discrimination between the top standard in A and the second standard in B. So it's actually developed very quickly. So what I'm going to do now is I'm going to stop the colour development. And the way that we do this is to denature the enzyme. And we denature the enzyme activity by using sulfuric acid. So in this pot, the 0.1 molar H2SO4 sulfuric acid we put in the separate trough, and we're going to add 50 microliters per well. So in the multi-channel, I'm going to put a tip onto each of the 8 wells, and once I've done that, I'm going to add 50 microliters into every well on the ELISA plate. So a bit fiddly putting all these on, and there we have it. So I'm going to take 50 microliters and denature and stop the colour development in each of these columns 1 to 6. And as I put them on, what you can see is the acid is also converting the blue colour to a yellow colour, and it is the intensity of this yellow colour that we will be measuring in a spectrophotometer at a set absorbance of 450 nanometers. So ultimately, in this ELISA, the intensity of the yellow colour is directly proportional to the concentration of the antigen that we are measuring. In the context of this protocol and this practical, we are going to be mimicking the measurement of CRP for inflammation or infection, and we're also going to add another scenario whereby we're mimicking the measurement of P24 HIV-1 antigen as a measure of HIV-1 infection. Okay, welcome back guys, and finally we've got the developed ELISA plate, and what we want to do is read it in the plate spectrophotometer. So you've transported it safely, take the lid off, and then what we're going to do is we're going to put the plate into the spectrophotometer, whereby well A1 goes to the top left in the plate holder. We put it in nice and gently, and there it's loaded into the spectrophotometer. Now what we want to do is we're going to read it at a wavelength of 450 nanometers. So with your mouse, you are going to select the green read button at the top and just simply click the mouse once to start it. And it says, would you want to replace previous data? Yes, replace previous data, click OK. And then what will happen is the plate will be taken into the spectrophotometer. This will read the whole plate from the intensity of the yellow colour that's directly proportional to the antigen. Finally, what will happen is you'll get a display of all your results. So what I want you to finally do is to get your phone, take an image, collect an image of all the results, and it is these results that you will use to set up the standard curve so that you can quantify the amount of antigen in test samples A to D.